This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 376. Today is February 12th, 2016. And yes, we are talking about Zika, but don't go away, there's some cool stuff. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from North Central Florida, Rich Condit.
Howdy, Vincent. How are you?
Happy Valentine's Day.
Thank you. Well, 2 days early, but I'm celebrating already.
Oh, it's Sunday, right. Yeah, you were out singing, weren't you?
I was out singing. We were singing Valentines.
So do you have a song you sing, or you just...
There's
a specific song?
As a matter of fact, we have a collection of songs that are appropriate for Valentine's Day. I think there's about 5 of them. Heart of My Heart, Let Me Call You Sweetheart, I Love You Truly, Love Me Tender, and Always. Okay? And anybody who buys a Valentine gets 2 songs, a rose, and a card and a picture.
Well, it's 4 guys, right? Barbershop, right? Who show up at the door.
Well, there are, yes.
4 old guys, too, right?
Now.
Now, I just don't know how that's very romantic.
One of our clients was in tears when we left today, and I think she was actually. I don't think she was in anguish. I think that she actually was,
All right.
Thought it was okay.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody.
Is it as cold there as it is here?
My goodness, it's way cold. It's -5 Celsius, 23, but the wind chill has been killing us the last 2 days. So I'm trying to find a Feels Like app. Feels like 10, although it's 23.
Yeah, the wind is bad. It's -3 here. Yesterday we did a TWiP with a guy in Montreal. It was -25 Celsius. Oh, my gosh.
Yeah.
I'm surprised it's that cold. It's 72 here.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. I was going to thank Rich for not doing a weather report from Florida, but he blew it.
Yeah.
Yeah, it is brutal here. When I was about to take my daughter to school this morning, I looked at my little digital outdoor temperature thingamajig, and it was saying it was 6 Fahrenheit.
Last night it was so cold in my house that the internet slowed down. Is that possible, by the way? Like the fiber optics freeze up?
It could crack, I guess, and stop, but I don't think it would slow down.
Also joining us from Worcester, Massachusetts, Jeremy Luban.
Hello.
I guess you're in eastern Massachusetts, right?
It's sort of central eastern, I guess.
Just barely, yeah. It's the usual dividing line.
Central, what would be the right appellation? Central Massachusetts?
Sure, why not? Or just Worcester. It's sort of unique.
Yeah, it's not Western Massachusetts, right?
No. We have to drive a little bit to get there.
So the miracle of modern transportation. Last Friday you were here in my office. Today you were in Worcester. Amazing, isn't it?
Yeah, I got away cheap last week. I didn't have to talk about the weather because I was in your office.
But you have the same weather as Alan Dove.
Yeah, it was 14 degrees in Boston this morning.
You were in Boston today?
Yeah. Now I'm in Worcester, and it's supposed to go down. Let's see, I forget what scale we're in.
Saturday night, it's supposed to be -10 Fahrenheit for the low.
Yeah.
Which is minus a gazillion Celsius or something like that.
It's going to go into it's supposed to be, someone said it's going to be like -25 centigrade.
That sounds about right.
So
I bet that's in real feel, though, not real temperatures, maybe?
No,
I don't know.
No, the National Weather Service.
No wind.
The forecast. The forecast absolute low temperature for Saturday night is -10 Fahrenheit.
Wow.
That does not count wind chill. There's also a wind chill warning, but just the absolute temperature is friggin' cold.
But again, Verkhoyansk is still, luckily, is still colder. It's going to be in the -40s or so.
So Jeremy is back because we're doing Zika again, which involves medical things. So we've got to make sure that we don't get anything wrong, right?
Yeah.
As if you would pick it up. No, I'm just kidding.
Exactly. I'll check the sodium and make sure.
Listen, last week you talked about Guillain-Barré, and none of us could have had that, experience. So.
Oh, that's true.
Yeah, All right. This episode of TWiV is sponsored by the 32nd Clinical Virology Symposium. I can't help laughing every time I read that. This is a meeting which is going to take place from May 19th through the 22nd in Daytona Beach, Florida, where you can bet it will not be minus 20 degrees Celsius. And you have until March 17th to submit your abstract to talk about your research on rapid viral diagnosis, the clinical course of viral infections, and preventing and treating virus infections. I'll bet there'll be some Zika papers there.
It's going to be a full second longer than last year's meeting.
Yes.
That's right. It's going to there are going to be 1,000 people at this meeting. They're going to be researchers and primary care physicians. And you can go to asm.org, CVS 2016, asm.org/CVS 2016. We thank ASM for their support of TWiV. And why I say there will be papers on Zika is because at the beginning of this week, I thought, well, let's find another virus to talk about on TWiV. And then all of a sudden, papers started pouring in. Well, maybe not pouring. A few came in, got letters, and some cool stuff happened this week. I really didn't expect it to happen so quickly, so here we are. Now, the first letter is from Anthony. It's a little bit of follow-up. It's more later. One of your guests questioned the travel warnings concerning Zika, noting that there are no alerts for France, where the incidence of toxoplasmosis is high. This was nice as a rhetorical flourish, but the comparison is not apt. The parasite problem in France is thought to be due to the consumption of undercooked meat, something quite easily for a travel to avoid. There are also screenings and drugs. Dickson should have been there. Well, he wasn't. Yes, he should have, but he was hacking out in the outer office. And so
and you don't mean on the computer.
No, although one could say what he does is hacking. Oh, I have to be nice to Dickson.
You have to be nice to
Dickson. I don't need a whipping boy, right?
No.
Glad someone answered me.
It took us a minute.
Jeez. So I sent it to Carolyn Coyne, who said, I don't necessarily disagree. However, one could argue that given that Zika is transmitted by a mosquito, precautions could also be taken to limit exposure. My point, as I'm sure you know, was to point out that these precautions are a bit of an overreaction. Pregnant women are tested monthly or for toxo in France, which we do not do in the U.S. given the much lower rates of infection. But there are no drugs that can be used to treat it. Yes, there are screens, but there are also therapeutics being developed for Zika, I'm sure. But I don't really think that screens taking place would make me feel any better as a pregnant woman who would travel. And you will see there's other issues as well later. I'm going to have Carolyn and her collaborator, Ed Boyle is his name, or Boyle, I know is his last name. I met him in Washington this week. They work on toxo crossing the placenta. So we'll have them on TWiP. That'll be fun.
Good.
Carolyn wants to be on all the Twix podcasts, and she said, "Can I get a card and I'll punch a hole in it every time on one, and then will you give me a sub at the end?" Or maybe
she's into a very interesting field. This notion of the placental barrier to infection is fascinating.
It's really good. It's understudied. And the coolest part is that she got into it while she was pregnant, thinking, I wonder why I don't get more virus infections.
The way she told that story at ASV is she and her technician, both of whom were pregnant, were sitting in the virus lab, culturing virus, and looking at each other and saying, is this okay?
Right.
Should we be doing this? And then they went to their... they went to a placenta biologist to talk to him about this whole thing and said, could we get some material to culture cells from a placenta? And he said, sure, bring a bucket.
Bring a bucket. Oh, yes, the bucket. And, of course, here we talk about 2-bucket infections.
Right.
I got the docs here talking 2 buckets. Now, I give a lecture about enteric infections every year, and I got them talking about 2 buckets.
But, making primary cell cultures from almost anything is a big deal.
Yeah. Yep.
But primary placental cultures, I'm sure it's not an easy task, but certainly the material that you need to do it is available.
But now, Rich, you get stem cells and you differentiate them. Or you get,
true.
And you can get any kind of cell you want. It's amazing. It is.
Yep.
It's just amazing.
Yep.
All right, so I got an email. I think this was Tuesday, on my way to Washington D.C. to do a TWiV down there from Arcan or Archan. I don't know how we would say it. Kathy, what do you think? Archan?
I don't know either. Sorry.
I'm a silent fan of TWiV. I've had a long-standing fascination with viruses, trained for a year as a Hughes Fellow at the NIH with Tay Zhang, and worked on our early understanding of HIV's tar interactions in the early 90s. I'm now on the faculty at Jules Stein, UCLA. The Zika story is fascinating, and the eye involvement goes along with findings we see in some other torch syndromes. The complexity of placental penetration from TWiV 375 notwithstanding, the virus is getting into the eye and destroying the developing retinal architecture, potentially suggesting wider neurologic deficits in these children, which will likely become manifest as they get older and are further investigated. Now, he sent me a link to a paper we'll talk about. I don't know why he concludes that the virus is getting into the eye because, I mean, as you will see, these are defects in children whose mothers may have had Zika infection, but you just don't know that that's Zika in there. There's no evidence for it. The eye happens to be a nice place to look because, as he said, other torch pathogens cause eye defects, and it's easy to look at, right? Not non-invasive. They have these instruments, right?
Right. Before we go much further, have we ever described what TORCH stands for? I don't think we did last week.
I thought Carolyn did, but you can describe it.
So
we did not.
The initials stand for toxoplasmosa. That's T. O is other. R is rubella. C is CMV, and H is HSV. And I think predominantly HSV.
CMV being cytomegalovirus, HSV being herpes simplex virus.
I just love the O. I think that's just a cop-out, isn't it? It's like I think Zika's in there with O, right? Or might
Absolutely.
be.
Well, it is. Yeah, it's going to be.
It will be. So did we say what O was, such as syphilis, varicella, mumps, parvovirus, and HIV?
Well,
but they couldn't put that in there because then it would be, it's
a cop-out. And then maybe it's going to be torches. Put a Z on the end.
Well, okay. So I want you to take all those things, including other, and make up a fancy acronym for it that we can all pronounce.
Alan could do that.
Alan, work on that.
Yeah, acronyms aren't really... that's my wife's thing. She's the crossword puzzle fanatic. I'll see what she can do.
And your thing is the clever titles, right?
Yes.
By the way, there was a tweet this week. Someone loved what someone said on TWiV last week. And this woman put up a picture of Boromir from Lord of the Rings and said, one does not simply walk into the placenta. She loved it. And I thought it was Carolyn, but it turned out to be Alan Duff, of course.
I came out with that, yes.
So you were thinking about Boromir saying...
I was.
What did he say exactly?
It's one does not simply walk into Mordor. And he starts this monologue about how hard it is to...
It's great. I love it. And then, so I didn't realize this. And finally, the fake Dickson said, "You don't know your memes, do you, Vincent?" Oh, well.
Back on TORCH, a TORCH screen is a blood test that's given to pregnant women. I guess it tends to be given when she may have had an infection. I guess it's not given routinely. Is that true, Jeremy? Do you know?
I don't know.
Oh, go away. Well, doctor here is useless.
Anyway,
it's really great to, this is the way doctors ought to behave in the office, right?
That's right.
Say, hey, doc.
Look on the internet.
I don't know.
Well, when you're waiting for them after they examine you, it's like a half hour before they come back. That's what they're doing. They're looking it up.
Right.
Okay, here's what it says. A TORCH screen is typically performed when a woman shows symptoms of any of these diseases during pregnancy. These can cross the placenta and cause birth defects in the newborn, including cataracts, deafness, intellectual disability, heart problems, seizures, jaundice, low platelet levels. So those are the defects.
But
they don't show they don't describe the symptoms, which I thought
insist on having it done anyway, even though it's not recommended for everybody.
All right. So this paper was published in the Journal of the American Medical Association. It's called Ocular Findings in Infants with Microcephaly Associated with Presumed Zika Virus Congenital Infection in Salvador, Brazil. So that's a good title, right? It's sufficiently uncertain. Presumed, associated with presumed. Those two words are good. Mm-hmm. And the senior author, Rubens Belfort, is an MD, PhD at the Vision Institute at the University of São Paulo in Brazil, and then all the collaborators on the paper also. And there's someone from Yale who is an epidemiologist, I guess. They let the epidemiologist go through the numbers. And this is basically looking at, they had 29 infants with microcephaly from this region of Brazil, Salvador, Bahia. I like Bahia because it's the name of my sailboat. And they did this study. Now, this is interesting. Right now, we're in the peak of the hype, right? And the study was done last December 1st to the 21st, where we know already there was this increase in this surge in microcephaly. 31 infants, and they narrowed that down to 29, and they had confirmed microcephaly. And the mothers, the mothers were not tested for Zika. They were tested for the torch pathogens, as you heard, and have negative for toxo, rubella, cytomegalovirus, herpes, syphilis, HIV. And that was all done by serology, but they didn't do any Zika because, of course, their infections were over. But they did have, the mothers all said they had symptoms that sounded like Zika: rash, skin rash, fever, arthralgia, headache.
80% of them.
80% of them.
23 of the 29.
79.3%.
So I have a question about this, because there's this and a couple of other studies that I've read. One was in MMWR, involved situations where patients were selected because the children had microcephaly. Okay, so that's the initial screen. And then they ask the mothers whether they've had symptoms of a Zika infection during their pregnancy. And in all cases where I've seen this, it comes out to be 70 to 80%. Now, of course, they're reporting symptoms that could be something else, blah, blah, blah. But the Zika is only symptomatic in about 20% of infections.
That's correct.
So this makes me wonder whether if you pre-select for kids with microcephaly, you wind up with a higher percentage of individuals. You select out individuals who had a symptomatic Zika infection.
Maybe.
Okay.
But again, a
thought.
Right, but the problem is still the missing denominator.
Sure.
If we got 30 randomly chosen pregnant women in Brazil and asked them if they had experienced these symptoms, would we get 80% of them saying yes?
Yes,
could be.
That's a problem, right?
And then these are, as you point out, they're very, very nondescript symptoms.
Right.
Particularly in an area with multiple circulating tropical diseases, and the screening is just it's what's available, but it's not really getting the statistics very well.
Yep.
Certainly, any kind of subjective report is going to be difficult to evaluate when the subject already has a traumatic medical experience. So,
yes.
I mean, the point here is that this is what we have to work with, right, at this moment. And
I would imagine that down the road, when we get a bunch more numbers, this will be resolved. But I thought it was interesting.
Yeah, I agree. I memean, it's early days, and a lot of these studies can be criticized in many ways, and we shall. But it's what we have to work with at the moment. And, you get some data, although it may not be the best. Now, of those 23 mothers who had the symptoms, 18 or 78% had them in the first trimester, 17% or 4 of them had it during the second trimester, and 1 of them during the third trimester. And they looked in the eyes of these infants, and you've all had this done. You go for an eye exam, and they put the thing on your face. They put drops in your eyes, and you can't see for a couple of hours, I guess. And they put a big machine on your eye and look at your retina, and you can see the picture of the retina on the screen in front of you, and they point out things that you can't recognize, but it looks red and veiny, And these kids had ocular abnormalities. 10 of the 29 patients with microcephaly had ocular abnormalities, 34%. Things like lesions, pigment modeling, atrophy, and so forth. If anyone would like to expound upon these, it's fine with me. But I think the point is
the eye exam is the only way you can actually directly view the brain in a person who's sitting in your office. So you actually can see the optic nerve, which is a direct extension of the brain. It's the only opportunity other than breaking open someone's cranium.
Cool. I knew we had to have you here for that.
So it's hard for most general docs to read, though with experience and time they get better at it. It's also kind of uncomfortable for most patients to sit through, but it's an incredibly valuable exam because you can look directly at vessels, blood vessels. You can see damage from all kinds of diseases like diabetes and hypertension. And apparently here you can see abnormalities actually in the optic nerve, though they don't tell us exactly what those are. Damage to the optic nerve suggests that there is something central going on that's going back to the brain.
Now that could be infectious or non-infectious, right?
I would think so, yeah. Anything, right?
So, Jeremy, when you're a medical student, do they teach you a little bit about this sort of thing, or is that too specialized?
Well, anybody, I think, who gets an MD in the United States should at least should know how to do this. It's not easy. I think most medical schools students actually have a rotation just looking at eyes. They spend maybe a week in the eye clinic and that's all they do. But it's something any good general doc, of which there are fewer and fewer on the planet because everybody's getting more and more specialized, it's something that any good general doc should be able to do.
Okay.
Well, and this is it's not just the drops in your eye annoying exam that you get at the ophthalmologist's office. This is also, you go to the doctor and they shine a light in your eye and look up, look left, and
yeah, I would say any thorough physical exam by a general doc should include a retinal exam like this.
Well, when I go for my eye exam for my glasses, they do this and they put a picture of my retina up with the optic nerve and the veins and everything. They do that now for an extra $30.
Yeah, you have a way high-tech doctor.
Really?
Yeah.
It's just Pearl Vision.
Well, I don't have that here at the university.
As I said, it's New York and San Francisco.
I get that usually.
Really? You don't?
Well, I'm thinking of this little handheld machine that's pictured in the link that I put in that also works.
I know how to use.
The ophthalmoscope.
Right.
Yeah.
So they say, when I go for the eye exam, they say, for $30, we can, and that's not covered by insurance. They say we can take a picture of your retina and see the nerve and the veins. So I've had that done for years for me and the kids, and they store them, so they compare them from year to year to see, and an old guy like me, if anything is happening, right? So it's really useful.
And so the little handheld device is an ophthalmoscope, and the process is fundoscopy because you're looking at the fundus inside the eye, right?
That's right.
Okay.
Very good. Very good. Now, so this is essentially the study, and it's just suggestive because there's many things missing. We don't know if these kids were actually infected. We don't know if the mothers were infected. So if you wanted to do this properly, it's hard to do in retrospect because the mother's infection is a long time before the kids are born. So basically you have to start at the beginning and have a cohort of pregnant mothers, screen them for infection, and then follow them through the pregnancy, screen the babies at birth, and then do this, right? Wouldn't that be the way to get really definitive information?
Mm-hmm.
Right. And I was also wondering, I don't think it's mentioned, maybe they presume everybody knows this, but are these normal findings in an infant with microcephaly?
Good question.
Yeah.
I mean, are eye problems typical of microcephaly, or is this something weird?
I bet you cannot find that information. Anywhere. There's probably a couple of people on the planet who know the answer to that.
Maybe one of them's listening. So please write in.
Well, the thing is viral. So with rubella anyway, it also causes microcephaly, but it also causes ocular defects.
Right.
So maybe they were thinking, let's see if this could be like a viral infection. But that's a good question because it.
Actually, I'll bet you Archan can answer our question.
Yeah, Archan is listening because
he's got to be listening.
He's probably thinking I should be there, and right. You should be. But answer us, Archan. Now, Kathy found another paper, right, that came out later in the week.
Yeah, somehow I was just trying to download the ones that you put in the show notes, and I got this other one instead. So this was written in a Brazilian journal, but it's totally translated into English, I guess. Or anyway, it's got a abstract in English and Portuguese. So quite similar because they have the caveat in the title, presumable intrauterus Zika virus infection. And they find, conclusion, I can't remember. They had 10.
They have 7 mothers.
7 mothers, 10 children? Oh, I thought it was 10.
7 mothers, 6 in the first trimester with rashes and arthralgias, early in the same thing. And then the kids' ocular findings,
17 eyes, which was 85% of the 20 eyes, which is from the 10 children. So, 10 infants, with microcephaly as defined by circumference more than 2 standard deviations below the mean, clinical diagnosis of Zika virus fetal infection. So I don't know how they got the clinical diagnosis of Zika virus fetal infection, but.
So they have 10 infants from 7 mothers, is that correct? So there
No, I don't think it's 7 mothers. I think it's 10.
Because in the abstract that PubMed, it says 7 mothers referred symptoms. Yeah. I don't know why, but you're right. It's 10 infants.
And then it says of 6, 6, which is 85%. Yeah, so somebody didn't proofread this.
But essentially the similar findings, right? The same kinds of ocular defects.
Mm-hmm.
Right. All right. So that on its own would not have merited another TWiV on Zika. But then the CDC published a report on somebody's phone. That's Jeremy Luban. I knew it because he doesn't know the TWiV routine.
I'm probably missing out on a cruise to the Caribbean.
I get Howard from the loan department.
Morbidity and Mortality Weekly Report, Notes from the Field, Evidence of Zika virus infection in brain and placental tissues from 2 congenitally infected newborns and 2 fetal losses. So I think this is a continuation of, I could be wrong. I usually am.
I think this was the story that we heard in the popular press that wasn't yet peer-reviewed and published.
Exactly. That's my assumption because it sounds so familiar. I mean, it sounds like exactly the same thing, and finally here it's published.
So in December, formalin-fixed paraffin-embedded tissue samples from 2 newborns born 36, 38 weeks gestation with microcephaly. They died within 20 hours of birth, so they had tissues from them. And then 2 miscarriages at 11 and 13 weeks. CDC got that from Rio Grande do Norte in Brazil. And at the time, they didn't do any virus probing, but now we have the results of that. 4 mothers had clinical signs of Zika infection, fever, rash, etc., but they didn't get tested for either virus or antibodies. So they did RT-PCR, targeting 2 different parts of the viral genome. Specimens from all 4 cases were positive by that assay, and they sequenced the PCR product and showed it was Zika virus. But, of course, it's a fragment, right? It's not the whole genome. They didn't try and get infectious virus out because this is formalin-fixed and paraffin-embedded, so you wouldn't be able to get virus out of that.
It is nice that they're able to RT-PCR it, though.
It is. Mm-hmm. And that's similar to what was done, by the way, for recovering the 1918 influenza virus from formalin fixed paraffin-embedded specimens way back from 1918-19, can be done. Let's see, in the newborns, only brain tissue is positive by RT-PCR. It's very interesting. So the virus seems to, if the virus is there, it would be neurotropic. Specimens from 2 of the 4 were positive by immunohistochemistry. They found viral antigens, so they have antibodies that they're using in what looked like mononuclear cells within the brain, and in another one within the placenta itself from one of the miscarriages. No dengue virus here. And they also saw histopathologic changes in the brain. So alteration in the brain cells, which would possibly be a sign of virus infection. And they also tested for the torches, and they were negative. So these are pieces of viral RNA and some viral antigen, which, again, it's not definitive, right?
Right.
Because
and again, we're looking at pregnancies that ended badly.
Right. So it could be other things, and it just happened to have some Zika fragments, and we don't even know if the whole genome is here. Who knows?
And one of the things that I've been wondering since this, I mean, other than the numbers question of is this the normal level of microcephaly, is this something where development starts to get derailed and the baby is going to have some kind of defect and that allows the virus to get through?
Sure, absolutely. Could be.
Or is this perhaps some kind of a situation with a cofactor that could be another virus, that could be something in the environment that permits Zika virus to get in? So it's very hard to determine what the percentage of blame is here.
Absolutely.
So it reminds me of HPV and proving that that's associated with cervical carcinoma and all the kinds of things that you have to do in the absence of having the ability to do Koch's postulates, take the agent and put it back into the individual and recreate the disease. Right. You have to have a lot of really good epidemiology. And so even if they found the whole virus in these, it still doesn't, it's not causation. That's
no,
say.
You have to do epi for sure, but having the whole genome is good. It's a good start. And here we don't even have it, nor do we have virus from anywhere.
But it is pretty amazing that of all the tissues they do PCR on, the only one that's positive is the brain.
Yeah, that's
you don't usually find flaviviruses in the brain.
Well, that's a good question. I mean, so we had a little conversation on email this week, and Carolyn said basically, who knows what viruses are in fetuses? We don't really look.
Who knows what viruses lurk in the minds of men?
Sure, but I think it's reasonable to assume that the fetus is
sterile.
Yes.
I don't know about that. Look here.
Yeah.
So I went back to TWiV 355, baby's first virome.
Yes.
Now this isn't definitive, but they took newborn and they had its first feces, and then of course they collected feces many days afterwards, and they just sequenced it. And at day 0 in the feces there are eukaryotic viruses. All right, so at least in the gut, and probably the baby's getting it from the amniotic fluid, I would guess, right? She's drinking the amniotic fluid. So if it's in the gut, who knows where else it's going? We have lots of viruses in our tissues. Maybe it's the same. Now, I agree the brain is a little bit extreme.
That's fishy.
That's fishy. But, Carolyn said basically we don't really have a lot of data on the fetal virome because for obvious reasons.
That's for sure.
For sure, right?
No,
but
that's true.
And I wonder how much we know about the fetal blood-brain barrier. I don't know much. I can
Probably
tell you that.
not a lot more than we know about the placenta.
Yeah.
I mean, considering how easy it is to get placental tissue and how little we know about that and how relatively difficult it is to get fetal tissue.
Right.
Yeah, this is all I think poorly explored territory.
Yep. And when we have something like this happening, then it stimulates more investigation, and we have tools now that we could get at this better. All right, so even after this, I mean, this would have been like a couple of follows up, but then a paper came out, and I think Alan had said this was under embargo.
Yes.
And it was about, what was this, Wednesday or Tuesday? Wednesday, right?
Tuesday. I think Kathy mentioned it.
Tuesday. I was told it would come out at 3, but in fact, I guess it came out at 5.
Came out at 5, and I get an email from the New England Journal every week with their table of contents, and I frankly almost always just delete it. Because it is so unlikely to have anything relevant to any of the beats that I cover. So I had missed this, and then Kathy mentioned it, and I logged into the press site and looked at it, and by that time, it was already Wednesday afternoon, so I figured, okay, well, this is going to come out open access in a few minutes anyway.
We've done a few New England Journal articles.
Yes.
Mostly they're case reports like this.
Yes.
The tick-borne high fever virus, for example, was a New England Journal and so forth. So this is called Zika virus associated with microcephaly. It's a single case report from Slovenia.
Yes.
Ljubljana.
Have you been there?
No. Have you?
Yeah.
Really?
Yeah.
The university?
No, not the university, but I've just been to Ljubljana as a tourist, and Lake Bled, which is just up the mountains from there.
Yeah, so this is the medical school, basically, in microbiology, immunology departments, gynecology, obstetrics, radiology. And it is a single case report, which I thought was pretty cool. And this is
beyond cool. This is chilling.
This is the best of these that has come out so far.
And if the rest follow the standards in this paper, then we're going to have a lot of...
I think these authors have said... The first author, by the way, is Zsernezs Mlakar, and the senior author is Tatjana Zupank, and I've probably butchered those, and I apologize. But,
that's why I didn't try them. And Jeremy, you've had some correspondence with one of them. Which one did you correspond with?
I wrote to, the corresponding listed was the last name.
Okay. Tatjana.
I wrote to her, and then I'm trying to remember. I received, my request was sent. We can explain what that is later. I don't know. I have to look up the email.
Okay.
Anyway, they replied immediately to a request I sent them, which was nice.
That's very good. I'm sure they're getting lots of requests.
Yeah.
All right. This is in October of last year, a 25-year-old European woman, healthy woman. She had been working in Natal in Rio Grande do Norte. Got pregnant in February, week 13, high fever, musculoskeletal retroocular pain, and a rash. Okay, so she didn't have a Zika diagnosis, but again, that's consistent with it. She went to Europe, had ultrasound, 14 and 20 weeks, and everything looked good. She went back to work, and then she went back to Europe at 28 weeks, and ultrasound showed some issues with the fetus, fetal abnormalities, and another one done at 32 weeks confirmed growth retardation and microcephaly, or apparent microcephaly.
And she reported that there was less activity in the fetus.
Less activity. So this raised suspicion of viral infection, and the doctor said this is not good for the fetus. So she said, let's terminate it, and they got approval to do that. And so she then gave permission for them to publish the results, which is really good. And this was determination at 32 weeks, so it's relatively late, right? Yeah, Jeremy, is that relatively late? Yeah.
Mm-hmm. Out of 40, yeah.
No, kids.
That's very unusual, right?
Full term is considered 36, is that right?
Yeah, it's still a little early, but yeah, it's awful. It's not a nice experience for the mom, let's just say.
No, I can't imagine.
Yeah, yep. So then they have the aborted fetus that they could study, and they did an autopsy, and it was definitely microcephalic. They did sections and examined the tissues, and there are lots of problems with this fetus, particularly in the CNS that they go into. There's calcifications, there are abnormal structures, lots of cells where they shouldn't be.
They do say, though, that grossly the fetus was normal in all respects, except
microcephaly, right?
Except for microcephaly, yeah.
They did sections of the brain and did electron microscopy. And you could see virus-like particles about 50 nanometers in diameter. And
that I think is a really
amazing.
That really caught my attention.
Yeah, that's the first evidence we've seen of real virus.
Mm-hmm.
Right.
That goes beyond nucleic acid.
Now, of course, electron microscopy doesn't tell you what virus it is.
Of course,
but it's consistent with what
with what they're looking for. Yes.
Yeah, it looks like it has an envelope, and so it's not a picornavirus for sure.
Right.
So it's an envelope. There are lots of envelope viruses, but back in the SARS outbreak, some of the first EMs said, "Hey, this looks like a coronavirus," because they have distinctive morphologies, right? They do some PCR, so they only say that they did EM in the brain, but I suspect they looked elsewhere.
Probably.
They don't report it. They do some PCR on the tissues, and they get PCR positivity for Zika virus on the brain tissue. And then they get...
And they looked at all the tissues. They looked
at
all
the
the organs.
So
and
only
they were all
brain,
negative.
Right. Only the brain is positive by PCR.
And they did PCR for everything.
The whole genome, and they got the whole genome.
No. I mean, for every flavivirus, for every torch pathogen.
Yes.
And everything was negative, but
except Zika.
Dengue, yellow fever, West Nile, tick-borne,
they did it right.
Chick, LCMV, CMV, rubella, VZV, herpes, parvoviruses, enteroviruses, toxoplasma. It's really well done. Amazing.
Yeah. And even the RT-PCR for Zika was not a single reaction. They did different, they amplified different genes.
Have a lot of RNA per milligram, 6 times 10 to the 7th RNA copies per milligram. That's pretty good. And then they were able to amplify the entire genome out of the tissue deep sequence, and they got the whole genome, 10,808 nucleotides.
Well, they did deep sequencing of all the RNA, all the reads, and they mapped them presumably against the human genome, but then also against the original Uganda sentinel monkey Zika virus.
Right.
And in those reads, of which there are, I don't know, a virus in a human sample like this, there's a million reads that would map to the virus.
So they took the RNA, Jeremy, and they have to make DNA out of it, right?
Right.
So they reverse transcribe it. Do they have to amplify it as well?
Usually to some extent.
There was a PCR step in this. So it's another...
Is that random priming?
Usually it is all random.
Okay.
Because you're looking for all the sequences. Right. You usually have to get rid of, depending on how you're making the RNA, how you're making the library. If it's total RNA, you have to get rid of things like ribosomal RNA, which are in vast excess, and make your sequencing very expensive.
This is all standard Thermo Fisher. They're doing ion torrent sequencing, so they use the Thermo kits for everything. The total RNA isolation. There is a PCR enrichment step again with a Thermo kit, which I think is random primed.
They're not amplifying with Zika.
Right. No, they're not specifically amplifying Zika in that process. So I think one of the things you're getting at there is that the odds that this is contamination are pretty low.
And Alan, you know this because you just read an article in Science about this. Right?
I just wrote an article about sequencing difficult samples, and in fact, one of the subjects within that article, and I talked to several people about, was sequencing formalin-fixed paraffin-embedded samples, which it turns out are among the hardest things to sequence in some respects, depending on the results you want to get, or the questions you're asking. But one of the nice things about formalin fixation is that it chops up the DNA into a bunch of little pieces. So you've got all this stuff fragmented, and that's what you want anyway for this type of ion torrent sequencing. You take little fragments and you sequence them.
But they didn't use formalin fixed in this case, right?
I don't know.
I think they had fresh.
No, I think they were frozen.
Yeah.
Were they frozen? Because I didn't.
It just says samples of tissue. It doesn't really say. It just says
because they formalin fixed the brain tissue and the spinal cord, and I don't know if they did that before sequencing or if they sequenced fresh tissue.
It says here RNA was extracted from 10 milligrams of all these tissues, cerebral cortex, trizoplast. So it may have been directly.
I think it's fresh tissue.
Yeah, fresh tissue.
Yeah. Okay. Yeah, they just say from the brain tissue. I think
fresh tissue samples were collected for microbiologic investigations.
Got it. Okay, yes. Right.
Hello, Dickson.
Hello, Vincent, and everybody else.
Welcome to TWiV.
Hi, Vincent. Hi, Dickson.
No, he's not Vincent. He's Dickson.
Yeah, I figured that out.
Yeah.
Welcome back.
Thank you.
We are talking about the New England Journal paper where they have just pulled out the entire Zika genome from the brain of this aborted child, and it is 99.7% identical with the Zika strain from French Polynesia 2013.
Wow.
And also a Brazilian strain from 2015.
Oh, cool.
Now, one thing I noticed from this phylogenetic tree is that if you compare those sequences with 1968 or 1996 Malaysia, 1968 Nigeria, 1947 Uganda. I can't tell how much, but there's been some drift.
Oh yeah, for sure. Yeah, they're the percentages there on the tree, right?
Yeah, you'd have to sort of measure it.
Well, they got the numbers for you, right?
Genetic distance in substitutions per site. I'd have to work on that, but
there is drift, and they see there's 5 polymorphisms in comparison with the Polynesian isolate, and they have amino acid changes, in specific proteins. So we don't know what they mean, and it would be hard to figure out because we don't have an animal model of this. But anyway, so I thought this was really amazing that they could get the whole genome out. They have particles there. It doesn't mean that there's infectious virus there, but it's pretty good.
Pretty good.
It's starting to add up. Yeah.
And it's only in the brain, which suggests it's neurotropic. So I wonder if they tried to get virus and were unsuccessful or what, right? They could take a little bit of their brain tissue and put it in cell culture.
Right. I have a couple of comments because we kind of glossed over the fact that because they had the whole brain, they did these sections of it and they show all kinds of abnormalities with respect to white calcifications and loss of gyration. So, of course, I had to look that up, and that's why I inserted these little figures in here for us. But gyration refers to gyri. Is that how you pronounce it, Jeremy? Yeah. Gyri versus sulci. And so those are the things that we think of as the wrinkles when you look at a brain. Or as a self-referential thing, like when you think of what brain coral looks like. Then, anyway, so
all of those things increase the surface area enormously and increase the cortical content.
Makes you smarter, Jeremy?
Well, I don't know if that is necessarily true, but it certainly gives you a lot more neurons to work with.
And it is definitely true that if your brain develops without the gyri, then you have serious problems.
Indeed.
Yeah,
and there's clear evidence of that here, and also the ventricles aren't normal, and so there's a lot of stuff.
Yeah, brain development is definitely messed up here.
So
then I was just going to say also I did check, and there is some literature about blood-brain barrier in development, and it's widely believed that in embryos and newborns it's immature or leaky, leading it to be more vulnerable to drugs or toxins entering the fetal circulation from the mother.
So I was describing this paper to my wife last night as we were taking a walk, and she noted that, according to my description, this woman, the mother, had a presumptive Zika virus infection at around week 13, and the fetus has evidence of a Zika virus infection, including particles and RNA, at week 32.
Yes.
Okay. So what does that mean? Does that mean, I mean, we don't know, but does that mean the fetus got infected when the mother was infected and the virus can persist in the fetus for that long a period of time?
Maybe.
Or does it mean that, in fact, there is persistence in the mother and the fetus could be infected later?
Yeah.
Okay. Right. I mean, we don't know, but I don't know.
Well, in rubella, the mother gets infected for a week or so, and during that time, the virus goes into the fetus, but the mother doesn't remain infected, as far as I know. Not that this is the same. I think you're right. Either way it could go. You can imagine the virus getting into the fetus early on and not having a great immune response. It could persist there, and there are lots of cells for it to replicate in. It's doing a job, right? It's really destroying it.
Yeah.
There's also the possibility that someone who is pregnant will have a much longer course of being viremic than someone who's not pregnant.
Right.
I mean, we don't know about that, but...
Okay.
Yep. Lots of questions here.
What is the immune status of a fetus? Can they mount an immune response?
Well, they get passive antibodies from the mother, right? Except IgM. Dickson, you just can't do that.
I can do it again if you'd like.
They get antibodies from the mother, right? They get IgG. And then I believe at some point, Jeremy, they make IgM.
Yeah.
That's what Carolyn said last week, anyway. Because it was.
Yeah, was she saying actually in intrautero they're making IgM?
Yeah, because they said.
Yeah, they can.
In some of these, I don't remember exactly, but they detected IgM in some of the fetuses, and that was. She said that's a strong indication that there's an infection in the fetus.
They do that
because the fetus would make the maternal antibodies coming into the fetus would be IgG.
Right.
Yes.
Right.
And so if there's IgM, it means those were made by the fetus,
correct,
most likely.
Yeah. That's the same story for toxoplasma, by the way. So I'm pretty familiar with that literature. And in order to know whether the fetus was infected or not, you'd have to look for the kinds of antibodies in the fetus and the mother. Right. And so IgM, for sure.
And of course, this all depends on where in development you are. If you're first trimester, then the immune system in the fetus is far, far less developed. And if you're, at 32 weeks, then obviously it's much further along.
I looked up something and found a story in Science News about the human fetal immune system arising from an entirely different source than the adult immune system. And this leads to some different reactivity to foreign things. The fetal system was thought to fail to see all those threats because it didn't respond to them, but these researchers at UCSF found that fetal immune cells are highly prone to seeing something foreign, but instead of attacking it, they allow the fetus to tolerate it.
Right.
This is a science article from UCSF researchers.
So for sure, the immune status of the fetus is, we can expect, is quite different than that of an adult.
Absolutely.
Right. And appears to switch over during the third trimester. And why and how that happens is not clear.
All right.
I seem to remember some kinds of experiments from the Racaniello lab a long time ago. Am I remembering correctly about challenging mice with viruses and certain things happening if they were neonates and if they weren't neonates?
Oh yeah, you do. You remember well. Better than I do. So we basically we have transgenic mice for the poliovirus receptor. One of the things we did is show if you infect a pregnant mother, the virus does not get into the fetus. It doesn't cross the placenta. Polio does not, for sure. The other one that you're referring to is we take newborn mice, transgenic for the polio receptor. You infect them with polio, wild polio, they get paralyzed. You infect them with the Sabin vaccine strains, they get paralyzed. If you infect a 3-week-old mouse with polio vaccine strains, they don't get paralyzed. So the only thing between you and paralysis is your immune system when you get these Sabin vaccines. Yeah. And there were people who were very unhappy about that result. I will not name them, but they said, they wrote, Racaniello is claiming that the vaccines are not even attenuated. And you know what? It depends what host you put them in to define attenuation. Because obviously in a 1-day-old baby, you can't give them Sabin vaccine. It would probably paralyze them because they don't have the immunity to deal with it. So that's the result you're talking about, Jeremy.
Yeah, then there was another experiment I remember from the 13th floor of the Hammer building at Columbia at the medical school there. That's where Vincent's lab is.
That's where I am, yeah. There's
another lab, Stephen Goff, who's been on TWiV, where they have a model where they challenge mice with Maloney-Murian leukemia virus. And the challenge is done with mice pretty much around the time that they're born. And if you take a whole litter of mice, you challenge them with Maloney-Murian leukemia virus, you come back 3 months later, they all have a malignancy that's associated with that retroviral infection. If you do the same experiment with an older mouse or a mouse that has a mature immune system, you never get that result, as I recall. So clearly, there is something to having a little experience to protect, or being a little bit older in your development. So there's lots of reasons to think that the situation for the fetus is very different.
Anything else on this paper? I want to move away from it. But before we do, let's make sure everything's okay. Yeah.
The only little tidbit which we referred to before was that there is an accession number for the sequence of this genome, meaning you can go on the internet and look it up. And I went to look it up, and the NCBI website where sequences are, and could not find it. So I sent an email to the senior author and got an email back from Misa Korva, who's one of the authors, sending me the sequence. I think it's an indication of how quickly they must have put the story together. So they've submitted it to GenBank, but it's as of yesterday, it wasn't yet.
Oh, cool.
So
my only comment on this, is that I hope people who are doing these types of analyses, and I know there are many more of these going on, will take this paper as their example.
Yep, for sure.
If you don't have access to electron microscopy or high throughput sequencing, then get a collaborator who does and start doing that.
Yep. And I should bring this up since you mentioned collaborator. I put out some feelers. I want to get some Zika and study it because we work on neurotropic viruses. And not everyone is giving out viruses and viral reagents. And I just want to say that is not the way science moves forward. You can't keep this for yourself. Science is not about you and your lab and your fame and fortune and making a lot of grants and getting a lot of people. It's about finding things that help humanity. So please share your reagents. It's kind of terrible.
But there is a little bit of leeway in that. It takes people time to grow up a viral stock and ship it out, and understandably, but the labs that have this are going to be slammed with requests. But other than that delay, you're right. There's absolutely no excuse for trying to hoard
people have
this.
said no. People have said no.
That's inexcusable.
It's ridiculous. Okay,
especially in a circumstance where people are suffering.
So
and need help.
So Vincent, if you Zika, you may not find.
Indeed.
Let Kathy.
Right. I want to cite the very last line of this New England Journal of Medicine paper from Malakar et al. It is likely that the rapid spread of Zika virus around the globe will be a strong impetus for collaborative research on the biologic properties of the virus, particularly since the risk of neurotropic and teratogenic virus infections places a high emotional and economic burden on society.
Yep.
Yes.
I wanted to just go through a few things that we don't know still about Zika. And you guys, this is all the way at the end. I'm sorry, but I'm jumping around. Like, why isn't there microcephaly elsewhere? We know there have been infections in South America and elsewhere. Why don't we see microcephaly?
And now, this came up, sorry to interrupt, but this came up last time, and I thought that a retrospective study on one of the epidemics in the Pacific Islands suggested that there was, in fact,
correct,
an increase in microcephaly.
I don't see the data. That's reported in some Washington Post or something.
The data on that are not out, and even if the data back up the reports, we're talking about very, very small numbers.
Yeah. It was not a big outbreak.
It was something like instead of 2 cases, they had 7.
Yeah, that's going to be a
Yap Island outbreak?
Yes, Yap Island, yeah.
It was either Yap or, I think it was one of those.
So and now we have a lot of infections in Colombia and other countries, and, I think they've been going on since the fall of last year, November-ish.
That we know about.
So we'll see what happens there.
Yeah, and I do think that I saw somewhere that they're tracking pregnancies in Colombia just for this kind of purpose.
Good. That's good.
Right. And it remains peculiar that Brazil had tenfold lower rates of microcephaly than the U.S. or Europe. I just want to remind people about that. And now they've come up to what may simply be the normal level. So we don't actually know what the reporting situation is.
Right.
Yeah. So the numbers are really hard to assess, and clearly they don't give you confidence that they have been recorded accurately. But they're dues, and you can look at the numbers overall for the whole country. You can look at the numbers by state, and then you could also potentially, and I haven't seen this, look by hospital with how many cases they're seeing. And I think that would be, if those numbers could be compiled I suspect from conversations I've had with people that those numbers would tell a story that the countrywide numbers don't. Just even if you don't know what the background was, what clinicians are reporting, again, it's subjective, but what they're reporting are really extraordinary numbers of kids within a hospital, which is kind of noticeable when there is a kid with such a disease. So
sure.
I think it's certainly conceivable that the numbers just ain't there, and they have to be obtained the right way to convince skeptics like us. The other thing in comparison, you have to remember Brazil is somewhat unique in terms of the timing of the arrival of the virus and in terms of the densities of the populations of the people. There are many more people in Brazil probably than in the regions of Colombia where the virus is infecting people. And it may just be a numbers game. They haven't seen
right.
An explosion in Colombia yet because it's too early, or the numbers are still kind of at the edge of where you would start to see the disease. So clearly not every pregnancy where you have infection is giving you microcephaly. So it may be a rare event. And, there are all kinds of interesting questions we could discuss, like is the virus in Natal different than the virus in Bogota?
Right. Well, what would you do with that, Jeremy? You can't do anything with that.
Well, at this point, no, but it is a possibility that there are different viruses. That information doesn't exist, and even if there were sequence differences at this point, we wouldn't be able to interpret them.
Right.
But as scientists, it's interesting to think that is a possibility. There could be variants.
I agree, and the problem is then the press says there's a mutation in the Brazilian strain, it's made it infect the fetus, and we just simply can't say that. Sure. I think scientifically I would love to know. And then you could look in culture and say, is there an interferon response difference because of this mutation and so forth? But yeah.
Right. No, it's total speculation.
Yeah.
Then there's also the host genetic situation. There are people who make a living out of finding rare, severe outcomes to common viral infections, for example, and tracking down host genes that are mutant in those individuals.
One of them is in New York at Rockefeller, right?
Right, Jean-Laurent Casanova.
I was thinking of that this morning, get some of these women and look to see if they have a polymorphisms that's predisposing them, and you could do cell culture to support it as well. Yeah, that's very interesting.
Well, and local level data, I think you raise a really good point with that, Jeremy, because it could tell you an awful lot, especially if there is some cofactor involved.
Right.
And not just an infectious cofactor, but
not just an infectious cofactor. I mean, people are proposing all kinds of things, and I know part of the control effort for any of these arthropod-borne diseases is going to involve a lot of spraying. I don't know how well regulated all that spraying is. And some of it may be with stuff that's not necessarily healthy for pregnant women to be around. Yeah. Now there's no evidence of that, but there's no evidence against it, and I think that's something that needs to be nailed down.
Dickson, are there more mosquitoes in Brazil than Colombia?
I don't think. Well, Colombia is a smaller country than Brazil, so I think the answer would be yes. But my question relates very much to the same questions that we had for West Nile, because you've got 21 different strains of West Nile. It's a similar but not the same virus, obviously, but it's the clinical symptoms vary from place to place, and they are dependent upon the strain of virus that you do encounter. So what you may be looking at here is a similar story with Zika with regards to adult presentation of syndromes. I was going to ask whether or not it's useful or not to know when to start to interfere with the transmission of Zika when there's a tip of an iceberg outbreak of dengue, because it's the same mosquito species. So maybe dengue is easier to detect because there's more symptoms associated with it. People are more used to it, and so therefore we could put an alert that, and by the way, Zika virus might be on the increase as well.
So something that we haven't talked about yet is this article by Fauci and Morens in the New England Journal of Medicine, A Perspective. And they mention that decades ago, African researchers noted that Aedes-transmitted Zika epizootics inexplicably tended to follow Aedes-transmitted chikungunya epizootics. They don't reference that, though, and I wasn't able to track anything down.
You know anything about that, Dickson? Why that would happen?
I don't. Also, I don't know whether or not this is a vertically transmitted virus infection. Do we know that?
Someone told... let me see.
In the mosquito, you mean?
Yeah.
In the mosquito, yeah. No,
I know in ticks.
It's not known, but Rift Valley fever virus is. So I did a podcast Tuesday about that, and the lady from Fort Collins said it is vertically transmitted.
Because that would confound it if it were transmitted vertically.
I mean, another interesting question is, well, we sort of touched on this, last week we touched on the issue of probably not all of these cases of microcephaly are actually real. They won't get reconfirmed. They're not all seropositive. So if you get infected with Zika, what is the chance that you will get birth defects? Now, we have some experience with other viruses. Rubella is a virus known to cause congenital birth defects. And there, when there were outbreaks, 1 in 1,000 infections, you would see a case of congenital rubella syndrome. So, I mean, the other thing that's kind of scaring people is that everyone who's infected probably is not likely to develop birth defects, although you don't know who it's going to be, right?
Sure.
But the actual frequency, we don't know. We don't know when during pregnancy is the worst time, right?
But we do know, I think we do know that this is not 100%.
Based on the limited serology that we have?
Well, because the distribution of Zika virus is so broad, there's a lot of this virus in Brazil.
Yeah. There is, yeah.
And you would expect a whole lot more. I mean, we wouldn't even be discussing whether this was a real problem, because it would be every kid born there would have microcephaly. But that's not what we see. What we see is these,
So I had written a blog post about this. What is today? Friday, earlier this week. And David Morans, who's at the NIH, wrote a letter and said, other flavies have in fact been linked to birth defects, but it's pretty rare. And he cited West Nile virus, Japanese encephalitis virus, and he says, if those viruses were really causing birth defects, we would see it because we have so many infections. And he said Zika is so close to dengue. This is bizarre. He said, something's going on. It doesn't all add up here. And he says the Zika viremia is lower than dengue by 10 to 100-fold, and so he's surprised that it even crosses the placenta at all. So I actually went in the literature and I found, there are a couple of reports on Japanese encephalitis virus and West Nile crossing the placenta and getting into infants and replicating there, but it's very, very rare. So, we would epidemiologically, we would know that if West Nile caused a lot of birth defects, right? I mean, it could be that in a very rare, it's rare for Zika to cause birth defects, but this is mixed up with something else that's going on. We don't know.
Some cofactor thing.
Yeah.
Here.
This perspective authored by Fauci and Morans that Kathy mentioned is really nice.
It's very good.
That ought to be put in the show notes.
It was actually out last week already. I didn't mention it, but David mentioned it to me when he emailed, so I thought, it is good. It's a nice, although as Kathy said, he doesn't reference that a couple of the things that would like to, and can't find it.
It is open access. I think everything.
It is.
There was just recently a discussion of this in the journals world that a bunch of prominent journals signed on to an agreement that if there's a public health emergency, they'll make all the stuff related to it open access.
Good. Can the scientists sign on to
make available everything?
So I also, on the blog post, someone posted, it's Fernando Spielke, who I've seen in the news talking about Zika a lot. I believe he's down in Brazil, but correct me if I'm wrong. He said the gross pathological findings, referring to the New England Journal paper, although new for the genus Flavi, are quite similar to those found for intrauterine infections of pestiviruses. Another remarkable coincidence with those far relatives in the Flaviviridae family is the possibility of transmission by contaminated semen. So pestiviruses are a member of a different genus within the Flaviviridae. So Zika is a flavivirus genus. The pestiviruses are animal pathogens like bovine viral diarrhea virus, border disease virus, and these can cross the placenta and cause infections and loss of newborns in cattle and sheep, and they can be transmitted in semen. This is a big issue when you use bulls to inseminate cows. You've got to make sure they don't have these pesties. So that's really
And if you use anything else to inseminate cows, then we don't want to know about it.
I was just going to say something about that, Alan, but thank you for interrupting.
So this led me to look up rubella, and as everyone knows, rubella is a plus-stranded enveloped RNA virus, but it's a toga virus, a different family. And back in the 40s, it was an ophthalmologist by the name of Norman Gregg who noticed that he'd see lots of cataracts in kids every time there was an outbreak of rubella, and these used to happen every 5 to 9 years. And eventually it was proven that cataracts, other eye problems, microcephaly, and deficiencies of many organs was caused by infection in the first trimester, the first human virus shown to be teratogenic.
Wow.
And because of that, a vaccine was developed, but it wasn't licensed in the U.S. till 1969. It has eliminated congenital rubella syndrome from the U.S., but it's not used in many parts of the world. And we have, according to WHO, 100,000 cases a year of congenital rubella syndrome in places like Africa and India, Afghanistan, Pakistan, because they don't immunize.
So while we're all bugging out about Zika virus,
I know.
Right.
There's a lot of kids being born with, and this thing is preventable because we have a vaccine, measles, mumps, rubella vaccine, which has eliminated it from the U.S., could eliminate it from these other countries. Yeah. So I don't know. I feel weird about that, you know, there are a few thousand cases. I know this is a bad thing to have, but there's lots more kids out there who are getting, for example, the last time there was an epidemic in the U.S. of rubella, 1962, 12.5 million cases of rubella, 20,000 children with congenital abnormalities. That's like 1 in 1,000 live births, something like that.
And this, I think, is something that ought to be discussed in the context where everybody's saying, oh, Zika virus, we need a vaccine against Zika virus. Well, here's an example of something that we know causes serious birth defects, is a serious problem worldwide, and we can't vaccinate against it.
Yeah,
We've got a working vaccine. So if we do get a vaccine against Zika virus a couple of years from now, after a huge effort, what are we going to do with it?
Yep.
Right.
Another interesting last point I'll make about rubella, the virus appears to infect most of the organs of the fetus as opposed to what we have in this one paper today. It looks like Zika is limited to the CNS, but we'll see about that.
Am I right in assuming that rubella is also the cause of SSPE?
No, that's measles.
That's
measles.
Nah, so I'm wrong.
Measles, but it's okay. We are all wrong. I was wrong today 17 times.
Really?
Yeah, for
Jeez,
sure.
I wish I was here to hear that.
You were teaching, weren't you?
I was.
This episode is also sponsored by ASM Microbe. This is their huge meeting that's going to take place in Boston this year. It's the fusion of their general meeting and ICAC under one roof, June 16th through 20th, Boston, Massachusetts, same state wherein live Alan Dove and Jeremy Luban.
Yes.
Jeremy, you still there?
Yeah.
I know Alan is coming.
Yep.
Are you going to go to this meeting, Jeremy?
I'll see about that.
I'll see about that. Well, we're doing...
You're even closer than I am, Jeremy.
That's for sure.
We're doing a TWiV and a TWIM. So,
or you could come to ASV, which is slightly overlapping, and afterward.
Yeah, I am going to that as well. And anyway, ASM Microbe is going to feature all the best in microbial sciences in the world, full scope of microbiology, new venues and programs, and there's going to be a session, for example, on microbial art.
Neat.
Including our friend Michelle Banks and the Vexed Muddler.
Cool.
Nice.
It'll be really cool. This is the meeting you should go to, if anything, to meet Alan Dove.
Absolutely.
Learn, connect, and advance at the inaugural ASM Microbe 2016. You should go to asm.org/ASM Microbe. There's 2 M's in there, ASM Microbe, to find out more about this meeting. And do come and say hello at TWIM and TWiV for sure. A couple of emails. The first one we've kind of touched on. It was sent by Robin, and it is an article in a website called The Ecologist where they're blaming the insecticide to cause microcephaly. But, of course, they have less they have less evidence for that than Zika.
Yes. And the reason I was kind of oblique about my mentioning of this possibility is that a lot of the sources that are promoting this hypothesis are full of hype, and they're anti this and anti that and chemophobic, and it must be the chemicals that are causing this. There are a lot of pesticides that are really not harmful to people and that really ought to be used to control the mosquito population. But a broken clock is right twice a day, and it is possible that there's something going on here.
I like that.
Right. It's evidently relatively newly introduced, and maybe that's...
Right. Could be, sure. I don't think we can discount it. We'll see. And the epi will really nail it down because... And
the claim here is that this pyriproxyfen was introduced in 2014, and it's been used apparently in drinking water tanks as a larvicide.
Oh.
Which, again, I'm not thrilled with the source that's promoting this concept, but I think this is something that is worthy of investigation.
So has anybody asked whether or not they've searched serum banks and things like that for how long Zika has been around in Brazil?
Not been done.
I have not
seen it.
But
I have not.
You should do it, though, right?
You can find all the liture on Zika in like 10 pages.
No, I understand.
The science journalist commented just the other day on Twitter that the weird thing about reporting on Zika virus is you can pretty much read the entire scientific literature.
I know.
Yeah,
it's very cool.
Yeah, but they should do that.
But Alan,
right?
do they understand it?
No, some do.
Some do. Alan, could you read Anthony's email, please?
Sure.
Sorry,
Anthony writes, as far as I'm concerned, the longer the better. If I need a couple of sittings to finish a podcast, that's great. Your podcasts have been a great spark for me. I've tried to start one with very limited success and have extended my blogging, and he provides a link to This Week in Birds.
That's great.
Which sounds like an awesome podcast.
Look, he's got a picture of a canary in a coal mine.
Coal miners with a canary. And,
that's cool. All right.
Very cool.
Anthony sends us lots of email. So check it out. Kathy Spindler, can you read the next one?
Okay. Carol writes, when I first heard the news about Zika and microcephaly, I immediately thought of the rise of anencephaly, I believe in the 1980s, along the Rio Grande. Then it was attributed to agricultural pollutants and others coming from both the U.S. and Mexico. One can't help but wonder if a viral disease was the real culprit. Any thoughts? And someone's inserted a link that the CDC has evidently investigated this, and they say lack of serological evidence for an association between Cache Valley virus infection and anencephaly and other neural tube defects in Texas.
Of course, that was 97.
Right.
They were looking at defects 74 women, 1993 through 95. So
and
anencephaly
after that was when Sin Nombre was found, right?
Yeah, Sin Nombre. Jeremy, what's anencephaly? No head?
No brain, I believe.
No brain.
No brain.
That's right.
Yeah. Well, it could be, Carol, but we don't know, right?
Hantavirus.
Jeremy, you want to take the next email from Spencer?
Spencer writes, dear TWiV team, the Zika episode was another great one for TWiV. Perhaps one of the best. I've been listening since the very beginning of time. No, he doesn't say that. Your discussion, we're going to talk about that time thing later. Your discussion centered on contracting Zika virus during pregnancy and the difficulties of crossing the placenta. Is there any information about the long-term sequelae of Zika virus infection in humans? Certainly other viruses and other infectious agents can have long-standing effects even years after the initial infection occurs. This leads to a question of whether a woman who contracts the virus, maybe even many years before pregnancy, is also potentially at risk later on, even if the viremia is not sustained. What evidence exists that this is or is not a concern? Thanks, Spencer Kroll. Mud fun.
Yeah,
I think no evidence, right? We don't know.
No, there isn't any. But of course, for rubella, this doesn't happen. You don't have any persistence, and it's an acute infection, and it's that time of the infection that the fetus is at risk. But it could be different for sure, right?
Haven't we had some small case report evidence that the mothers are no longer positive for the virus?
Yeah. That's why they don't know the status of the mother, right? They can't do PCR, right?
Right. So that might
virus, actual direct assessment of the virus is difficult. There's I think out about 5 days is as far as you can find it in the blood, and apparently the urine is maybe out to 10 days. That is by RT-PCR. So it seems like it gets cleared pretty quickly in most people. But then there's all the unknowns, like do you make antibodies against Zika virus that later cross-react against fetal cardiac tissue, or anything's possible. So
or dengue.
Or dengue.
We have a couple of articles sent in by Tim and Anthony about this young lady who downloaded all the science papers that have been published and have put them online for everybody to look at. And, I agree that papers need to be open access, but you can't really do things that are illegal. I don't think that's a good idea, right? Am I being too conservative here, or you probably should go at it through other channels?
This is a very, very old argument from many fields well outside the open access world.
Just steal things and make them free.
Is it? Well, there's the property issue, and there's the civil disobedience issue, and
All right.
Is it right to obey an unjust law?
We went through all this with the music industry several times.
Yeah, we did.
Oh, the music industry, the movie industry.
So Apple fixed the music industry, so maybe they could fix publishing. Hey, Rich Condit, can you?
I think the only thing that's clear is if you do it, you have to be sure you don't get caught.
Well, she's talking about it. She's not. I mean, she's like Edward Snowden. She's saying, "I did this," right?
Yes.
Unless that's not really her.
Aha.
Ah.
All right, now it's too complicated for me. Rich, can you read Vic's?
Sure. Vic writes, Dear TWiV team,
TWiV.
I'm a long time listener, episodes 1 through 374, and a first-time emailer. I thought I should say something regarding Alan's remarks concerning cruise ships and norovirus. That was episodes ago.
Yes.
I should tell you that I live in Kristiansand, Norway, and work as a ship's agent for one of the world's largest suppliers of logistics to the cruise industry. And listens to TWiV. How about that? This is great.
Yeah.
When a cruise ship arrives in a new port, the first person on board in the morning and the last person to leave the ship in the afternoon is the ship's agent. The agent handles paperwork for the police and customs, arranges transport for ship's personnel, purchases supplies, finds spare parts, and most importantly, coordinates medical services for crew and passengers. I am on board more than 100 different cruise ships each season here in ports along the southern and western coasts of Norway. Alan is correct that most cruise ships are flagged in countries that require fewer regulations. Here in Norway, we see ships that are primarily flagged in the Bahamas, Malta, and Antigua. However, the regulations that the ships are trying to avoid have more to do with taxation than sanitation. There are very strict rules concerning labor laws, environmental contamination, and especially sanitation for vessels that call here in Norway and other European ports. The cruise ships are, without exception, completely paranoid about sanitation. There are hand sanitizing stations at every gangway, at every elevator and stair landing, at the entrance and exits to every restaurant, in the crew galley, and in all areas where passengers gather. Some of the ships have personnel stationed at the gangway who require you to sanitize your hands when you leave or return to the ship. The ships desperately want to avoid the terrible publicity that is generated when 350 passengers share a bout of norovirus, so they are as proactive as they can be to minimize the potential of an outbreak. I don't think they are so concerned with the publicity generated by taking advantage of tax breaks from Malta. Love the show. Keep up the good work. Medvenlig Hilsen, which Kathy has translated for us as with best regards. Vic.
Point taken.
Mm-hmm. However, alcohol hand sanitizing stations are not going to be as effective against non-envelope noroviruses as just plain soap and water hand washing.
So are they not effective at all? Is this all a waste of time? Or I guess this covers bacteria, fecal contamination, right? This is better. It's better than nothing.
Probably better than nothing.
And it, helps for influenza.
Yeah.
But for noroviruses, I think you really need soap and water.
They can't do that up and down all these gangways.
Right.
And I think I may have mentioned this before in this context, but one of the things that has always been missing in this whole discussion of cruise ship norovirus is what are the rates of norovirus outbreaks in resorts that are on shore, like all-inclusive resorts?
Oh, I have that data for you. The
Really?
cruise ships are really a minimal. Most of the outbreaks, 90% of the noro outbreaks, are in catering, restaurant, institutionalized setting.
Well, right, yes, it's the most common foodborne illness.
But the cruise ships are a small fraction, but they get all the publicity.
Right, but I think the relevant comparison is to all-inclusive resorts because you have everybody together in those too, and I suspect the rates are probably quite high.
Yeah, I don't know about that, but there are.
But that's not tracked because they're actually in other countries.
Yeah.
So evidently the most effective hand sanitizers maybe reduce norovirus counts by 2 log reduction. Friction seems to be really important.
Friction.
Mm-hmm.
I think you should wash your hands with vodka. And then when you're done, you could take a drink and you won't know if you get sick.
And then at least the projectile vomiting will be clear.
Yes.
True.
Dixon, you don't have a reader, right? So you can't read.
Sorry.
Okay, no problem, Dixon. I don't have a problem at all, actually.
Oh, good.
No problem.
I'm glad to hear
He's
that.
being nice to you.
We have 2 left. AJ writes, "Hello, TWiV team. I read what was to my friend an alarming article and to me a very funny one, and he gives a link to a really just a terrible article about what's causing chronic fatigue syndrome and all this. Sorry if you have already discussed this, although there isn't much about the article itself that's really worth talking about." And he's right, except that Epstein-Barr virus actually likes cheese. And there is a line in this article. So the idea here is that, most people are infected with EBV, and he's saying when it's reactivated in certain people, it's causing chronic fatigue, which is not bad. But then he says things like the virus bides its time until it senses stress-related hormones. Yeah, okay. And where's the cheese part? I lost it. All right, I'll search for cheese because I have to read this to you. Here we go. You develop sensitivities to foods that never caused you problems before. This happens when the virus consumes a food it likes, such as cheese, and transforms it into something your body doesn't recognize.
Oh.
I didn't know EBV liked cheese, did you? He continues, makes me wonder if it prefers a good bottle of Pinot Grigio or Malbec with its cheese. But after reading this, I was delighted by the humorous aspects and rather horrified how it could be very seductive to people. Blame problems on some external enemy. I tried to think of what people could do to combat such evangelistic misinformation, and I realized that what you guys are doing is probably the best cure. Talking to people about medicine in an honest and thorough way and making it understandable. So thank you again. Although in the article's defense, I will thank the medical medium that his cure is to eat healthier, and I guess that probably will help people with a lot of their problems. Thanks, AJ in San Diego, where it was enviably warm degrees Celsius today and sunny.
I would suggest that anyone who likes cheese not look at electron micrographs of cheese. It's quite disturbing.
Have you done?
Well, I think the viruses generally have their cheese with fava beans and a good Chianti.
No, I heard an amazing talk. I'll try and track it down. It's incredible, the microbiology of cheese. All of the creatures that make a really good cheese look pretty scary under the electron microscope.
Cheese is so wonderful, isn't it? Does everyone here like cheese, or do some?
Love cheese.
Oh,
yeah.
It's wonderful.
It is.
It's just microbiology at its finest, isn't it?
Well, the wine isn't bad either, and that's microbiology.
That's microbiology too, yeah.
Sure.
Yeah, the ASM has put out one of their booklets. I think it's one of those Academy-produced things about cheese.
Yep.
There were articles in the Microbe magazine for a while, and then it all got collated into this booklet that we have out where I eat lunch, and I've been working my way through it.
You remember when we did TWiV 300 down at ASM headquarters?
Good.
That was part of the Microbes After Hours.
Yeah.
They had one on cheese, and Chris Condian said it's the most popular episode they've made.
Yeah.
People like to know how their cheese. All right, one more email, and it's Alan's turn.
Okay. Justin writes, also just curious, is there any merit to purposefully giving a woman Zika before she tries to get pregnant as an attempt to avoid microcephaly? Links to an article and frequently asked questions from the Washington Post.
I don't know what this has to do with that.
I don't know what this...
Yeah, but this would be a parallel with before there was a rubella vaccine. I just remember when I got the German measles, my mom was happy, as a young child.
Right, like chickenpox parties, and that was the...
right.
Well, and going back even further, I guess, a variolation.
Mm-hmm. Of course, there was a high fatality rate.
High fatality, yeah, all of these things had high complications.
I could not recommend that because we don't know any of the long-term sequelae of Zika.
No, we don't. Right. I don't think there is any merit to purposefully giving anybody Zika virus at this point.
What do you think, Jeremy?
I think if there were a similar virus that you got from milking cows and it protected you from Zika virus,
you've been reading too much, Jeremy. Much too much.
Yeah.
Hey, Jeremy, these notes at the end are yours, right?
Yeah, I just thought I was kind of amazed. I'm new to this pro-med mail thing, and
cool.
Just over a couple of days,
it'll give you medical students disease all over again.
Right. Anyway, I thought it might be worth. This is just a couple of things I picked just from the past couple of days. So there's a yellow fever virus outbreak in Angola, and as best I can tell, they're on the order of 40 people dead since December.
Yep.
There's a little girl, 11-year-old girl in Argentina who seems to have died from Junin virus and another arena virus. Venezuela, apparently 60 people have died very recently from Guanarito virus. And in Nicaragua, apparently there's been a lot of howler monkeys found dead without any obvious trauma or any other obvious cause. Kind of sounds like a virus. Apparently somebody actually wrote in wondering if that could be...
Yeah, Anthony sent us an article saying, is Zika or something else killing Nicaragua's monkeys? Who knows?
And then finally there's this little outbreak of Lassa fever where in Nigeria there's 176 cases and 108 deaths. This is according to the Minister of Health there. So there's a lot of virology going on.
And you can hear about it here on TWiV. And from people who work on the viruses, and you can get our own personal experiences. And I know it's long, but you could break it up, right?
Mm-hmm.
Hey, and today we're going to come in just a little over an hour and a half.
Yeah. Well, yeah, depending on how long Dickson's pick takes.
We aren't done yet.
Right.
Actually, I'm going to give your pick from last week, Dickson.
Good.
Because,
Good.
You asked me why we didn't pick it.
Yeah, why didn't you pick it?
Because if you're not here, you can't make a pick.
Oh, that's...
Like, who's going to talk about it?
That's fair.
Because you're the advocate, right?
Yeah, you want to...
I walked into the room and I didn't find a seat, so I had to leave.
Wait, let's ask Jeremy if that's true. Because he was here.
He was here. He says, oh, here's Dickson. I remember him saying that.
I take the fifth.
I'll join you on that.
Dickson, what is your pick?
I'll bring the glasses.
What is your pick?
Well, I picked the German fusion machine first. That was last week.
Sorry, I've got an autoplay going on. There we go.
Last week I picked that because they actually achieved fusion. But this week is the 25th anniversary of the Hubble Space Telescope.
That's this one, right?
Yeah. So I thought the pictures were remarkable then, and they are still now. When you look at them all at once, it's quite an awesome accomplishment.
I think it's pretty cool that Angela Merkel is a physicist.
Yes, and she got to push the button that actually started the machine that generated the electricity. So there you go.
And it doesn't leave a whole lot of radioactive stuff.
None.
Coming out the end.
If it shuts off, it just shuts off. That's the end. That's right. That's exactly right.
But your pick is the Hubble thing.
Yeah, I'll pick the Hubble.
All right. Alan, what do you have?
I have a handy little explainer from PhD Comics, and I think we've had a few PhD comics that have been picked. This one is explaining gravitational waves, which have been much in the news.
Oh.
Sorry, I muted myself so I could sneeze.
Gee,
Right.
Can you show Dickson how to do that?
Just, come on.
Yeah, so this is just I think a really good explanation of what the big deal is in this new result demonstrating the existence of gravitational waves, which proves that Einstein was in fact correct.
Yep.
And that goes hand in hand with my pick, which is the Times article on that.
Hey, by the way, that was mine today, too. So.
Yeah, but
Which has an autoplay video connected to it.
That's true.
Yeah, I know, but I picked it first.
How would I have known that?
If you had read the show notes.
Yeah, well, that's right. Oh, there.
But let's
okay.
not bicker, Dickson.
So I would drink to those suggestions. I would say this could be the most important publication, which was published yesterday about the gravitational waves in our lifetimes. It's really huge in its implications.
That's true.
Okay.
And by the way, Einstein actually didn't believe it. He did his best to prove that it wasn't true.
Right.
Well, that's the best science, isn't it?
Yep.
I actually have a second pick.
Uh-oh.
The Amazon Web Services is a division of Amazon. They have a lot of cool things, including storage, which I use, and other online programs, not having anything to do with the stuff they sell on their main website. So it's called Amazon Web Services, or AWS. And they issued a new terms of agreement, which I went and looked at. And paragraph 57.10, it says your use of these materials is not intended for use with life-critical or safety-critical systems, such as medical equipment, transportation systems, blah, blah, blah. However, this restriction will not apply in the event of occurrence certified by the U.S. CDC or successor body of a widespread viral infection transmitted via bites or contact with bodily fluids that causes human corpses to reanimate and seek to consume living human flesh, blood, brain, or nerve tissue, and is likely to result in the fall of organized civilization. I just had to pick that. It's so funny.
Mm-hmm.
I wonder if you are like the only person in the known universe who's gotten that deep into the acceptable use policies.
This has been widely retweeted.
Yeah.
Okay.
Yeah, I heard it on NPR.
It's really funny that somebody at Amazon has a sense of humor, and it's
somebody writing the legal document has a sense of humor. That's what's weird.
Yeah, lawyers having a sense of humor. Oh, I'm sorry. Some of our listeners are lawyers. Yes. Kathy, what do you have?
I picked the tileapp.com, and I was telling you guys about it last week at the beginning of the show. We still haven't run through all of my Christmas and birthday presents yet, but this is one that I got from my younger brother. These are Bluetooth-enabled chips. They're about a 1/4 inch, and then they're about an 1.5 inch by 1.5 inch square. And you can hook them onto your keychains. You can put one in your car, attach it to your camera, and then you set up the app in your phone or your iPad or your computer, and you can double-click on the. You can find your phone with these things, and your phone can find your tiles. So if you have your tile on your keys and you've misplaced your keys and you have your phone, you can find your keys, and vice versa. And it will find your phone even if you've got the sound turned off. It'll activate it, and your phone will make sounds. And if you get far enough away, then there's a map to help you find where your item is. And then if you've totally lost it and you can't find it, you can click on something, and then everybody who has this app on their phones in a crowdsourcing way is helping identify the locations of lost tiles.
Or who stole
that
it.
information gets conveyed up to the cloud, unbeknownst to the person with the app, and then the cloud will come to the person with the missing tile and say, your tile was last seen at such and such location, and then you have a chance of finding your keys or whatever. So they're not very expensive. You can buy one, you can buy a set of 4, you can get a bulk price and get 8. And
how many do you have, Kathy?
I got 4. And so, for those people who know someone who's always losing their keys or is misplacing their phone, it's really useful.
You could hook it onto your kids.
Yes.
Yeah. Most people have said to me, but this would be too big to put on my glasses, right? So, it would be a little awkward.
They're on the top of your forehead, Kathy.
Yeah. But somebody mentioned even like going to Disney World and giving each kid a tile.
Yeah, for sure. That would be cool.
How much for your car to find out who stole it?
Well, if you can't remember where you parked in the parking deck, because you park in, every day or something.
You could leave one in your car, right?
Yep, I have one in my car, but I hardly ever drive, so I can pretty much remember where I park. But
that's very cool. Excellent. So you got this for Christmas?
Mm-hmm.
Nice. Mm-hmm. I like that. Rich, what do you have?
So I mentioned this last week when we were discussing genetically modified mosquitoes, but didn't have time to go into it. In the process of educating myself about these mosquitoes, I ran across a video in what is JOVE, the Journal of
Visualized Experiments.
Right. Journal of Visualized Experiments. It's called Mass Production of Genetically Modified Aedes aegyptis for Field Releases in Brazil. And it's about a 20-minute video that shows you how they mass produce these mosquitoes. And I just found it absolutely fascinating.
And it's open access.
Yes, I believe so. And
not all Jove videos are.
To me, the best part about it is how they separate, because I was waiting for this, because they specifically isolate for release out of a mixed population male larvae.
Right.
And I was amazed at how they do this. You got to watch the video. I could describe it, but it'd take too long.
Rich, the lab that I was associated with at Notre Dame, run by George Craig, pioneered all those methods, and he could separate at the pupae, the females from the male pupae, based on their width.
Now, this is basically based on their size.
Yeah, right. Exactly. Exactly.
You pour them down between two glass plates.
Yeah, that's his method.
And form a wedge.
Exactly.
And you can see bands of these things.
Got it.
And just dilute them out the bottom. It's amazing.
Yeah, it's fun.
So I thought that was a cool video.
Somebody this morning emailed and asked for the link to this because they heard it on last week's and thought it was cool. So
well, there they go.
It's coming. Speaking of mosquitoes, I heard recently that there's a thing called the Microsoft Research Institute.
Go on.
It's a research. I am Dickson. Just relax. Okay. There's a research institute funded by Microsoft, and one of the things they want to do is make a real-time map of the world showing where mosquitoes are carrying certain viruses. You go to a website and you can see, on 91st Street, there's a mosquito in New York carrying some virus. So what they have designed is a mosquito trap in which the mosquito flies in, and they have it fixed up so that the mosquito flies in, and then the mosquito is smashed and embedded in paraffin, and then it's delivered and picked up by drones. And then these are picked up, they're sent back, they're deep sequenced immediately, and you have a map almost real time of what mosquitoes are carrying. So apparently these guys are willing to talk about this. We have to get them on TWiV.
This sounds fun.
Isn't this cool? I'm trying to get them on TWiV. Oh man, that'd be so cool. I'm sorry to digress. We are up to who? Jeremy.
Jeremy, I think.
What do you got, Jeremy?
Oh, well, there were a couple of things I was thinking of, but in light of the discussion about Zika, I thought it might be interesting to mention a paper from Jun Hu, H-U-H, that came out in Science, where he works with a mouse model where moms, while they're pregnant, are challenged with some sort of inflammatory stimulus like poly-IC, or it can also be done with viral challenges. And they find, reflecting an observation apparently that comes from the clinical literature, they find that this does increase the frequency of what are called autism spectrum disorders in the pups. So they have some kind of way of quantifying this in the mice that are born. So this is a publication in Science that just came out, and it specifically addresses the immune mechanism, and it involves Th17 cells in the mom that are somehow required for transmitting this effect on the pups. So I just thought it was interesting in light of all of our discussion about Zika and some of the mail that people have sent in with questions. There, of course, are all these questions that are wide open. Short of something like microcephaly, are there other potential sequelae that kids would suffer if their moms were infected with Zika during pregnancy? So,
cool.
Interesting.
Very interesting.
We'll leave it there.
That's it.
Okay.
You had another one, but you're not going to.
Well, I was going to mention a book that I started in anticipation of the announcement. I had been following the LEGO project that just yesterday announced that they had indeed found gravitational waves. And this is a book that I started really last week. I'm about a third of the way through it, and it's wonderful. It's called The Perfect Theory, A Century of Geniuses in the Battle Over General Relativity. It's by Pedro Ferreira, who is an actual practicing general relativist. And it's very accessible, and it talks about the implications of the general relativity. And the really exciting thing about it is that it points out that we really are only now beginning to come to terms with it 100 years after it was described by Einstein. And that there are many, many people now who understand it, something that was not the case, say, 30 years ago or 20 years ago. And that it's very exciting to have all these people studying it and working on it and now finding gravitational waves.
Cool. It's very cool. All right. That will do it for TWiV 376. You can find it at iTunes and also at microbe.tv/TWiV. We love getting your questions and comments. Please send them to TWiV@microbe.tv. Jeremy Luban is at UMass Worcester. Thank you, Jeremy.
You bet.
Maybe we don't need another Zika next week, right?
Who knows
It comes out on Tuesday, Wednesday, and Thursday.
Who knows?
Yeah.
We'll see. Probably. Yeah. I mean, I don't know if people are going to get sick of it, but this was an important week to do it again, right?
Yes.
I think so. I think the papers that came out yesterday are in New England Journal. That one case report really is a game changer, I think.
Alan Dove is at turbidplaque.com. You can also find him on the Twitter. Thank you, Alan.
Always a pleasure.
Kathy Spindler is at the University of Michigan in Ann Arbor, Michigan. Thank you, Kathy.
Thanks. This was fun, and I learned a lot.
Rich Condit can be found in Gainesville, Florida. Thank you, Rich.
Sure enough, always a good time.
He is an emeritus professor at the University of Florida in Gainesville and also a well-known barbershop singer. What do you call that? Barber pole? No, barbershop quartet.
Sure, barbershop quartet.
He's famous because of that.
Well, I wouldn't go that far, but
Dickson Despommier is here in my office at Columbia University, where he is an emeritus professor.
Indeed.
Thank you, Dickson.
You're welcome, but I'm not a barbershop quartetter.
Do you sing?
I do, but only by myself and when no one else is listening.
But you do play the trombone.
I do.
All right, very good. Someday you should play it for us.
No,
I say no, right?
I think you guys probably wouldn't want to hear this.
I've heard you play, actually, and I've recorded it.
Uh-oh.
I could broadcast it.
You wouldn't dare do that.
I have to be nice to you.
Be nice to them.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us, and we will be back next week. Another TWiV is viral.